Jaimo and Dr. Elia Di Schiavi for the organization of this webinar focused on the evolution of brain cortex. A special thanks for having kindly accepted the invitation for a talk to Nereo Kalebic from Human Technopole, to Daniel Del Toro Ruiz from University of Barcelona, and to Cristina Pirosi from Max Planck Institute of Psychiatry. They will drive us through a journey into mammalian brain cortex and its structures from evolutive, cellular, and molecular points of view, adding some pathological implication for human brain cortex. And now, to continue this very brief introduction, I have the pleasure to introduce you Professor Marco Moracci, Vice Director of the Department of Biology at University of Naples, Federico II. Please, Professor Moracci.
Thank you very much, Professor Lucini, for this introduction and the scientific introduction to this meeting. I bring the greetings of our Department of Biology that virtually is hosting this meeting, because one of the organizers, the chair of the meeting, is my colleague, Professor Di Jaimo. But really, it's a little bit funny every time we have to connect through webinars, because this is not really, it is a common place, it's not our department. So I hope that next year in December, we will be able to host you in our department in person, or at least partially in person, in a merged way. Our department is rather big. It's one of the biggest departments of biology in Italy. It's more than 100 people, and there are many different thematic issues that are studied here. So one of the topics is the object of this meeting. And I first of all, I have to thank my colleague, Rossella Di Jaimo, and Elia Di Schiavi, my previous colleague at the National Research Council of Italy, who were able to organize this webinar. So I think it's very important that we keep these communities together, also in very difficult times, and trying to find the advantages of having the possibility to keep in touch, maybe more easily without any flights and so on, even if we lost a lot, that is the personal contact. I forgot to say that I'm here instead of Professor Ricca, who is the director of the department, who was involved in other business. He made the tough work. I have the funny work in introducing these meetings, so it's my pleasure. So I wish that you have, and there will be, of course, a very interesting meeting. I want to thank the speakers of being here, and I hope you spend a very interesting afternoon discussing science on these very nice topics. So I think now the word goes to Rossella for the chairing the meeting. Thank you.
Okay. Thanks, Carla. Thanks, Marco, for the kind introduction. And good afternoon also from my side. I'm Rossella Di Jaimo, and I will chair this webinar today together with Elia Di Schiavi. So we are member of the scientific committee of the Napolitan Bay Group. And as Carla already mentioned, we organized this meeting in December with the idea to keep alive, even in these difficult times, the NBG tradition of a meeting in this period of the year. And we will have our next meeting, hopefully in person, in May 2021. The topic of this webinar is the evolution of the brain cortex, and I want to thank again Cristina, Nereo, and Daniel for kindly accepting my invitation, and we are looking forward to hear about their exciting stories. So now some technical infos. For the people who follow the webinar on Teams, please turn off video and microphones during the meeting, not to interfere with the presentations. And during discussion, you can turn it on again for questions. Every talk will last 40 minutes, and then 10 minutes for discussion. For every question or comment, you can raise your hand in Teams, or you can write on the chat on Teams or on the YouTube channel. Please, Elia, do you want to say something?
I think just, of course, I want to welcome all the speakers and thank the participants for being here. I just want to end that also on YouTube you can raise your hand, actually make questions, adding on as comments on your right on your right side there is a chat and you can make questions. I don't think we need anything to add, and possibly we can start with the first speaker, which will be presented by Rossella.
Okay, then the first speaker of today is Nereo Kalebic, that studied molecular biology at University of Zagreb in Croatia. He was immediately interested in neurobiology and did his master thesis at the Croatian Institute for Brain Research. In 2008, he moved for PhD to MBL to the lab of Paul Empestal, where he studied the role of microtubules post-translational modification during development and the function of the peripheral nervous system. For his postdoc, he moved to the MPI of molecular cell biology in Dresden in the lab of Wilhelm Huttner, where he studied the development and evolution of neocortex. Since September, Nereo moved to Milan, where he became a group leader, congratulations, in a new Italian life science institute, the Human Technopole. Nereo's research focuses on molecular and cell biological mechanisms underlying human neocortex development and its implications for human evolution and neurodevelopmental disorders. Today, he will talk about neural stem cells and neocortex expansion in development and human evolution. Dear Nereo, the stage is yours.
Thank you, Rossella, very much. I will first try to now share the presentation, and please let me know if everything works fine now, if you can see it. Can you? Okay, great. So thank you very much for the invitation, and to everybody in the Neapolitan Brain Group and the Department of Biology, and also thank you very much, Rossella, for this wonderful introduction. I'm very happy to take part in this, and I'm only very sad that this is not happening in person, because this is one of the best times of the year to visit Naples. So I'm going to talk today about neural stem cells and neocortex expansion during the development and evolution. And how I came to the neocortex expansion is through getting interested in how we get to be so intelligent compared to other species. So when we ask ourselves this question, we really need to focus on our brain, and in particular to the part of the brain called neocortex. This is the outer sheet of our brain that it covers roughly this entire area, and it is called neocortex or new cortex because it's in evolutionarily most and it is a seat of a plethora of higher cognitive functions such as language, but it's also controlling many of our, we can call it more basic behaviors through our various motor and sensory centers. So in spite of such really fundamental functions of human neocortex, we actually know surprisingly little about how it develops. And this happens during the fetal development and continues postnatally with a characteristic human feature of brain maturation that happens throughout our teenage years. And basically only in our 20s we reach full maturity of our brain. But the period of human development that I'm most interested in is the phase of this expansion, which is the end of the first trimester and the beginning of the second trimester, where neural stem cells and neural progenitor cells divide massively to generate more of these stem cells and then more neurons. And this is the period when the brain will start having its characteristic shape and first faults will also appear. So this is a key period for our development, and if something goes wrong during that time, the consequences can be really severe. And this brings us to malformations, so the changes in size or shape of the brain that can then result in very severe intellectual disabilities. And then a very known example of this is microcephaly, where the entire brain is reduced in size. And a few years ago, we had another virus that was very problematic for humanity, which was Zika virus, and it was causing microcephaly. We are more interested in those disorders that have a genetic origin, but Zika virus is one of the most known recently. A part of neocortex expansion during development, we can look at it also through the evolution of human brain. And interestingly, if we compare our neocortex, its size, to the one of the chimpanzee, which is our closest living relative, we can see that we have 3 times bigger neocortex. And this is completely disproportional to the body size, which is quite similar, actually. If we even look closer in our evolutionary history, we can see that certain species of Homo still had this 3 times smaller cranial volume than we do. And that actually only very lately during our evolutionary history, there was a massive expansion in neocortex size. And only us and the closest species, which are Neanderthals and Denisoans, are considered to have the brain of this 1 liter. So if we now put all this together, what we are really interested in studying is what are the molecular and cell biological bases that regulate this neocortex expansion. And we look at that through development, through evolution, and when something goes wrong in these molecular and cell biological mechanisms, we see how it can cause severe pathologies in humans. So to study these questions, we of course use human brain as our most important model, but obviously for experimental reasons, we also need different model systems where we can test functionally these genetical and cell biological mechanisms. And for that, the species that we use a lot is the mouse, which gave great contribution to neuroscience research and in general biomedical research. But when we talk about the expansion of the neocortex, mouse is not the most ideal model system unless we use it for gain of function studies. Because as this image here shows, the size difference and also the difference in complexity is enormous. And that's why we use also an intermediate model organism that recapitulates better some of more complex features of the human brain. And this is Ferret, which is a small carnivore, roughly the size of a cat. And it's a model animal genetically accessible, importantly for us. So if we make a cross-section of these brains like this, then it can immediately be clear to you why I think Ferret is an interesting model, because it so nicely recapitulates one of these classical characteristics of human brain, which is folding. Whereas mouse in wild-type conditions will not. But I'm sure during the next talk, we are going to hear wonderful data on how it can be induced. So we use all these 3 organisms to understand the neocortex expansion, and we use them at cell biological level. So to dive into the cell biology, I will show you first this developing neocortex of mouse at the embryonic stage 15, where neural progenitor cells are labeled in magenta. So neocortex develops as a classical epithelial tissue showing apical basal polarity, with the apical side facing the lumen of the lateral ventricles of the brain and the basal side facing outwards towards the skull. So if we zoom in the column like this, we can see that in mouse, I divided the neural progenitor cells into two largest classes. Apical progenitors, which are the ones that maintain contact with the ventricle, and basal progenitors, which are the ones that delaminate and they lose contact with the ventricle and move more basally in a new germinal zone called subventricular zone. So if you look into these two types of progenitors in these three species, we can see that apical progenitors remain more or less the same in terms of their abundance between mouse, ferret, and human. And indeed, they are also similarly proliferative in all mammals, as far as we know. But basal progenitors are the ones that show striking differences in their abundance between these species, with obviously human developing neocortex having the greatest numbers of basal progenitors, which suggests that these are the cell types that we should look in order to understand human neocortex development. And why are there these differences? So the differences stem mainly from the different proliferative capacity between basal progenitors in mouse versus ferret versus human. So I'm going to talk a lot about the proliferative capacity, and by that I mean an ability of stem cell to divide and generate other stem cells as opposed to differentiated cells. And basal progenitors in mouse have a very low proliferative capacity, meaning they will typically divide only once and give rise to 2 neurons. Whereas in ferret, and most notably in human, basal progenitors can undergo many cycles of divisions. They will generate more of themselves, and finally they will then generate many more neurons compared to ferret and mouse. So the key question that I've been interested here was to understand what are the cell biological differences that underlie this proliferative capacity between basal progenitors. And what we have found was that simply the morphology of these cells is likely one of the key contributors, and I'm going to tell you a bit about that story soon. But what we're also interested in is understand what were the consequences. Once we do increase the proliferative capacity of basal progenitors, how does this lead us to neocortex expansion? What are the mechanisms? And we saw that the specific increase in type of neurons called upper layer neurons when we achieve this. So I'm gonna start with telling you a bit about how morphology of basal progenitors we think is a key cell biological mechanism underlying their proliferation. So in terms of morphology, basal progenitors can come in 2 flavors. They can be multipolar, like shown here on the left, which are the cells that have a high number of small cellular processes, or they can be radial, and this type is called either basal radial glia or outer radial glia. And the morphological characteristic is these long radial processes that were thought to be only apically or basally directed. So we did a very simple experiment, and we labeled developing human neocortex tissue in culture with a dye eye, a lipophilic dye. And thereby we labeled all the cells that exhibit a contact with the basal lamina, including all the cells that have this long basal processes. And we found cells that have 2 basal processes, either growing from the cell body, like shown on the cartoon, or a single basal process that then bifurcates into 2 branches, as shown on this rotation. So I got really interested in that because the idea of having two basal processes is something that can lead us to thinking that if each daughter would then inherit its own basal process, it could then be able to maintain contact with the basal lamina, which is rich in pro-proliferative signals. And this might be a way to maintain stemness through generation of these progenitors. So I was really interested in that, and we looked at it in more details by asking the question if the morphology can actually affect the proliferation of basal progenitors. Can it be causative? And in order to address this question functionally, we had to establish several tools first in these model systems that would allow us to do it. Most of them, or almost all of them, are based on a new way of genome editing mediated via CRISPR-Cas9. And we applied it some 4 years ago to mouse system in vivo during the embryonic development through a surgery called the in utero electroporation, where the pregnant female is undergoes a surgery, and nucleic acids or proteins are injected into the ventricle of the embryonic brain, and then electrical field is applied to direct these molecules into the part of the brain that we are interested in, which in my case is neocortex. And we made the proof of principle of this approach by targeting GFP in a mouse transgenic line that expresses GFP in a subset of progenitors. And as you can see in this image in the middle, where there is a hole, that's where we achieved the knockout of GFP, and this corresponds to the electroporated area. We developed a similar way also to perform epigenome targeting in vivo. But as I told you, there are features of neocortex development that we wanted to study and that mouse doesn't have. So therefore, we were very lucky to have an excellent collaboration with Hiroshi Kawasaki in Japan, who pioneered the method of in utero electroporation in Ferret. And then I learned this method from him and brought it to Europe, where we can now do this. It's very similar as in the mouse, but since the animal and the brain are much bigger, it allows us to study some of these more complex features, such as folding, that you can maybe see on the second image. And we have just published a Jove article on this, so you can see an entire movie how this is done. But although Ferret is really useful for understanding in vivo some of this complexity of the brain development, there are features that are truly human-specific, or primate-specific at least. And these cannot be modeled in carnivores or rodents. So for that, we do use also fetal human neocortex through wonderful collaborations that I had in Germany with clinics. And we can also do ex vivo electroporation of this tissue and perform genome manipulation ex vivo in human tissue. And here is an example of how progenitors can be labeled with GFP using this method. So once these methods were established, we were to apply them to address the question of how morphology could be linked to proliferation. And we did that by performing in vivo labeling in mouse and ferret of basal progenitors and ex vivo in human. And here is an example of human basal radial glia, which we then tracked using image analysis softwares and performed quantification of various morphological parameters, which interestingly, most important turned out to be the most simple, which is just the number of processes. And by processes, I mean these cellular extensions that grow from the cell body. They, if we just look at the number of processes in multipolar progenitors, we see that human cells have more of them compared to ferret and mouse. And even more interestingly, if we now look into the basal radial glia, and we can see that human neocortex contains a larger proportion of those morphotypes that have 2 and more processes compared to ferret, and both human and ferret have more compared to mouse. If you look more into detail, you can see that these two morphotypes that were newly identified with two basal processes that we originally saw in human tissue, we could also recapitulate in ferret. They existed there, but we could not see them in mouse, suggesting that there were some quantitative differences between human and ferret, but the differences between those species on one hand and mouse on the other are qualitative. And it is one of the reasons to use ferret for some of this research. So when we got these very exciting interspecies differences, which we can also mimic as evolutionary differences, then we were really then interested to understand which genes could be underlying these morphological differences. And to address this question, we looked into all the genes that are expressed in the germinal layer where basal progenitors reside, which is called subventricular zone. And we focused on those genes that were strikingly enriched in that compared to other zones to have a specific transcriptome of basal progenitors or the histological layer where they reside. And from there, we narrowed to those genes that have a potential morphoregulatory role based on geoterms or data in literature. And then we picked one gene that also had interesting interspecies differences, and its name is palm delphin or palm D. So it's from Greek, brother of palm, and palm is paralemmin, a protein involved in synapse. And what this evolutionary difference was is shown here. Palm delphin in all mammals comes in two isoforms, but not both are expressed in all tissues. So if we look into the developing brain, we can see that both cytoplasmic isoform, which ends with this KKVI motif, and the membrane-bound isoform, which at its C terminus has the characteristic CUX box, both of them are found in human developing neocortex, but the membrane-bound one is not found in the mouse, which made this isoform immediately more interesting for me, and we sought to express it in mouse and ferret in vivo. And we also made a CRISPR-Cas9 mediated knockout of PALM-D in human neocortical tissue in culture. What these experiments showed us was that when we look into the multipolar progenitors, we can see that when PALM-D is added to the mouse neocortex, the number of processes increases. Whereas when PALM-D is knocked out in the human neocortex, the number of processes is reduced. And this is this showed us that PALM-D can change the number of processes of multipolar basal progenitors. But similar phenotype was observed also when we looked into the basal radial glia. But here we compared it to ferret because in mouse, basal radial glia are very, very rare. So when PALM-D is added to ferret, developing neocortex at embryonic day 35, the number of or the proportion of basal radial glia that contain two or more processes, so the green ones, is increased compared to control. On the other hand, when we knock out PALM-D, we specifically reduce this morphotypes with an increased number of processes. So this in total tells us that PALM-D is a potent inducer of new processes and has likely important morphological role. And what we now wanted to see is how could this be actually connected to proliferation, which was where this evolutionary component resides. So we did one of the most simple experiments of checking for proliferation, which is just counting cells that are cycling, or in this case, positive for Ki67. And as I told you, in the control mouse case, most progenitors are apical progenitors and basal progenitors are very rare. But when we express PALM-D, then the number of basal progenitors is massively increased, which is quantified here. It's basically doubled. In contrast, when PALM-D is knocked out from human tissue in culture, then the proportion of cycling cells in the subventricular zone is reduced, suggesting that PALM-D can affect also the proliferative capacity of these cells. But how are these two things connected? So we have a molecule that is known to bind to the membrane, and we have data suggesting that it could have an important role in establishing morphology. And we also have data saying that this can have a consequence on proliferation, but how is this connected is not still clear. So we went back to this membrane attachment to see whether really this is required, or perhaps PALM-D could have another role in the cell. And to address this, we used both the COX isoform, the membrane-bound one, and the other one, the cytosolic isoform, KKVI, together with a truncated construct that completely lacked this last protein coding exon. And what we saw, interestingly, was that only the membrane-bound isoforms could increase the number of processes and could increase the proliferative capacity of these cells. The others were not able to do this. So we next pulled down PALM-D and performed proteomics to identify some possible binding partners. And we found, and we did this in developing human, ferret, and mouse neocortex, and we picked those candidates that were enriched in human and ferret, but were absent in mouse, in order to find those that could be important for the membrane-bound isoform. And we found as the top candidate adducin 3, which is an anchoring protein that anchors actin cytoskeleton, depicted here in gray, with a plasma membrane. We also found that palm-delphin is palmitoylated to the plasma membrane, and this palmitoylation is also important for its function. So we then we think now that these molecules are making so-called morphoregulatory complex that is in a way inducing and promoting induction of these new processes. But to show whether to examine whether adducin 3 is actually having the same phenotype as PALM-D and whether this could also have influence on proliferation, we repeated the key experiments as with PALM-D, just this time with adducin 3. So we made a CRISPR-Cas9 knockout in human tissue in culture, and we examined the number of processes and the number of proliferative basal progenitors, and we saw that both of them are reduced upon the knockout, suggesting that these molecules are operating together. But again, this told us a lot about how processes are potentially built in these cells, but it did not tell us how we come to proliferation, because neither adducin nor PALM-D were found to have any role in proliferation, which is independent of their role of inducing processes. So we then started thinking differently and imagined that perhaps PALM-D per se didn't have anything to do with proliferation, but that it is actually this new processes, just mere presence of new processes that promotes proliferation. And that would mean that this cell now has contact with new territories that before it didn't have. And that if in these territories it finds new extrinsic signals that are pro-proliferative, this could explain why it is promoting proliferation. And basal progenitors in the developing human neocortex, they live in this stem cell niche of the subventricular zone, which is maintained by many different factors of growth factors, neurotransmitters. Serotonin has recently been shown to play a role there. Many factors that can contribute, but one of the main that have been known for a while are components of the extracellular matrix. So they make the matrix of this niche, and they were known to promote proliferation. So we sought to look into integrins, which are the main receptors of the ECM components, and to see whether they could be involved in PALM-D-mediated proliferation. So we did this experiment in the mouse, and we used the knockdown of integrin beta 1, which is the most expressed beta integrin in mouse neocortex development. And we did this knockdown together with overexpression of PALM-D. So on the left images, you can see how PALM-D produces this characteristic effect of increasing the abundance of basal progenitors. But when integrin beta 1 at the same time is knocked down, this effect is lost, which is quantified on the right. So if the hypothesis which I was depicting before was true, and if proliferation was merely a consequence of the increase in the number of processes, then manipulation of integrins that are regulating this proliferation is not expected to affect the number of processes, because this is something that should be upstream. So indeed, when we looked into the morphological parameters, the number of processes was as high as when integrin was not knocked down. It only depended on the overexpression of PALM-D. So with this, we set up a hypothesis which we last month published, and it's based on the fact that extrinsic signals of basal progenitors are the key factors that promote their proliferation. And we symbolically have shown this by using this specific type of jellyfish, which has two large central tentacles, very similar to basal radial glia with two basal processes. And they also have a lot of these small processes, which female jellyfish actually uses to collect sperm and uses that for reproduction. So it reminded us of how basal radial glia is maybe collecting its pro-proliferative signals from environment, which are needed for its own proliferation. And it's also a very lovely creature, this jellyfish, so we also chose it for that. So with this, I hope to show an interesting story of how morphology of basal progenitors might be one of these key cell biological features underlying the proliferative capacity of these cells. And now I'm going to slowly transition into the second part, which is how what happens next when we increase this basal progenitor proliferative capacity. What are the consequences on the brain development? And for this, I chose a gene for which I suspected would have a very strong effect on basal progenitor proliferation. This is ARHGAPIIB. It's a human-specific gene, and it evolved as a duplication of the ancestral ARHGAPIIA after the split of the human lineage from the chimp lineage. And it's shared by us, Neanderthals, and Denisoans. And interestingly, both us and Neanderthals have the similar size of the brain, suggesting that maybe ARHGAPIIB could be implicated in brain size. It was originally put in context of human fetal neocortex development by Marta Florio from Huttner lab, and found to be expressed in apical and basal radial glia. And when it was added to the embryonic mouse neocortex, it could increase proliferation of basal progenitors and occasionally induce also folding, which puts it in the implies it basically into the evolutionary expansion of the human neocortex. So I took this gene and I added it to the developing ferret neocortex because we wanted to put it in a more complex system to study this more longer term or medium term at this stage consequences. So we did the in utero extraporation of ARHGAPIIB, and as expected, it created a really strong effect on basal progenitors in ferret. Here we count the number of mitoses, and we can see that number of basal mitoses increased almost fourfold, which is an incredible increase during development. So it was really interesting to see what the consequences would be. This is the developmental trajectory of ferret, indicating the period when neurogenesis is happening, which is towards the end of the embryonic development and then continues also for a few days postnatally. When we added ARHGAPIIB at embryonic day 33, just before the peak of neurogenesis, what we caused was an extension of the neurogenic period. Likely basal progenitors that became more proliferative started dividing and generating more basal progenitors, and then generation of neurons was protracted with time in time. And we chose P16, postnatal day 16, as the date for analysis, for main analysis, because that's the time when neurogenesis and migration of neurons should be done in both control and ARHGAPIIB expressing ferrets. So this is a beautiful brain of P16 ferret, where you can see this lovely folding pattern. And you can also see the left hemisphere, which is electroporated and labeled by GFP then. So if we zoom into the cortex of these animals and into the upper layers of the cortex, you can see immediately, if you look into the GFP channels on the right, you can see that there are more green cells. These green cells, these are the progeny of the in utero electroporated progenitors. And but not only that there are more in ARHGAPIIB compared to control, but that there are also present more in the upper part more. So this is because cortex is built in the inside-out fashion. So the newborn neurons will bypass the existing ones and find their final position on top most basally. And these, therefore, the layers of the neocortex are built from the deep layer 6 and 5 to the upper 4, 3, and 2. And as you can see on these images, when ARHGAPIIB was expressed, there were more cells in the upper layers, more neurons, as is shown here with staining for SADB2. And there is just increased density of cells in general, which also led to the, in the end, increase in thickness between these upper layers, between control and ARHGAPIIB. So these features that I just listed, these are really the key hallmarks of the evolutionary expansion of human brain, because not only will human developing neocortex contain basal progenitors with a greater proliferative capacity than ferret, but also when we look later in development into human cortex, it will contain more proportionally more cells in the upper layer neurons compared to lower. And this is particularly also interesting for these callosally projecting neurons that were marked by SADB2 on the previous slide. So this is something that we are also following up on to understand how the basal progenitor increase in proliferation will then lead to an increase in upper layer neurons, and how will this be then translated further through development. So these are the questions that now I'm, as Rossella already said before, that I'm now studying in my new lab, which is we're just setting up here at Human Technopole in Milan. And we are interested in understanding the molecular and cellular mechanisms that are involved in neocortex development and in pathogenesis. We are continuing to understand the mechanism of how the process of basal progenitors is growing, which are the other molecules implicated in this, and how we can manipulate this further. But also in the context of human neuropathological diseases, and which especially here at Human Technopole is going to be one of the pillars in research, to understand how genomic changes that in basal progenitors specifically result in human intellectual disabilities or human neurodevelopmental disorders in general. Unfortunately, we don't know much about this yet, because most of the studies were done in mouse, where I told you basal progenitors are not so dominant. And mutations that were created in mouse could not fully recapitulate all the human phenotypes. And one of the reasons was probably because basal progenitors in mouse have such a low proliferative capacity. They just make 2 neurons. So if you any potential mutation there would not have such a dramatic consequences as in human, where the much more is at stake. So we are looking at this using human models, using Ferret also, and looking at it through the morphology to try to understand the mechanisms of some of these diseases. And as I told you before, we are also interested in going into more long-term consequences to understand at the animal level how, of course, then their behavior will also be affected once we increase the number of upper layer neurons. So at the end, I'd like to thank my postdoc advisor, Wieland Huttner, and everybody at the Max Planck Institute of Molecular Cell Biology and Genetics, where all this research that I showed you has been done. Wonderful services and facilities of the MPI CBG. And also now I'm here at Milan at Human Technopole. We are a group of 3 only at the moment, but we are expanding. And I'm sure in a year we're going to be many more. But we already started. And I'm now setting up also collaborators in Italy. So I'm also thankful for this opportunity to present my work to you, and I hope we can definitely remain in contact, and we can also set up some interesting collaborations in the future. And I thank you for your attention with this image of my home country, which I very much miss currently. Thank you.
Thank you, Nereo, for sharing your data. There's some echo. Okay, so then if there are questions, it's time to raise your hand. So there is Ennio del Giudice.
Okay. A compliment for, as a clinician, I would like to compliment you because this was really a wonderful presentation and very nice and very interesting to me. Even if I cannot go deeply inside your work, but your presentation was so clear and so attractive that I am, let's say, I am forced to raise my hand, even only for curiosity. Okay, the part that obviously interests me more is the part where the genes underlying the functions of basal progenitors are involved or are in or could be compromised in some brain disorders. So the question is just for curiosity. So first, if you know of other labs in the world that are studying, that are following your same research, and ideally, in which type of pathology you think that these genes involved in the functions of basal progenitors may be damaged, and if something is already, if there is already some speculation about what could we get from these studies. Thank you very much, and my compliments.
Okay, thank you very much, really, for your very kind words. In terms of diseases, first of all, let me say that yes, there would be a lot of labs that would be studying this type of diseases, and also the role of basal progenitors in those diseases. But I think that this concept that the morphology could be one of the causative factors so far hasn't been explored by others, with at least as far as I know. So I think this is something where we have a lot of possibility to find new mechanisms that were so far unknown. And in terms of diseases, I think these are mainly malformations. So those that would affect the size of the brain, like microcephaly or macrocephaly, so too small or too big brain, and the shape of the brain in terms of folded or not, so lissencephaly, polymicrogyra. These are the diseases that could be the most affected by the morphology of basal progenitors for two reasons. morphology affecting proliferation will affect the number of neurons produced, hence microcephaly, macrocephaly. On the other hand, morphology of basal progenitors, these actual processes are used as tracks for migration of neurons. And that is very important in lissencephaly or polymicrogyra, when this migration might be impaired, not because of the neuronal phenotype, but because of the fact that tracks, the processes of basal radial glia are not properly developed. And these are the diseases that we are most interested in. But since I know also that you work on the disorders of corpus callosum, so this is something that that is, for example, also one of the becoming interesting to me, since we see that these callosally projecting neurons are actually the most affected as a consequence of the phenotype in basal progenitors, and how could this then affect the generation of corpus callosum itself? It would also be interesting to follow up. You're muted, sorry.
Thank you very much. Thank you very much, and also for this mention of disorders involving the corpus callosum, which is a very, very nice structure, and what it is difficult to understand is in people with only agenesis of the corpus callosum, who can function generally quite well, even in school, and with no important, let's say, disorders. Maybe your studies and similar studies may contribute to better understand what is the problem underlying these functions. So thank you very much.
Yeah, thank you. This definitely it's our greatest hope. Thank you.
Nereo, I also have a question. It's more a curiosity. So you said relating to the first part of your talk, this PALM-D. So that PALM-D is increasing proliferation because it's working like sort of a sensor of an extracellular environment, inducing the cell to sense more the environment to more processes.
Yes.
Now I was wondering, do you have an idea or a hypothesis based also on your transcriptomic data, which is the way how this signal is translated to the cell, giving rise to more proliferation? So how this signal is translated, through which pathway?
Thank you. Indeed, we studied this. So we think that main extracellular signals could come from extracellular matrix that are then received by integrins. And then we followed the intracellular signaling, and we found that phospho-ACT, so PI3 kinase phospho-ACT pathway was upregulated, and also the phospho-ERK pathway. And then we were more interested in the phospho-ACT and the subsequent mTOR pathway, because these are the ones that are recently being shown by several groups to be very important in basal radial glia proliferation, and also implicated in human diseases. So we so far think that might be the most important intracellular pathway based on both ours and others' data.
Okay, thanks. And there is also another question from Daniel. So nice talk. And then he was wondering whether cortical folding in the ferret changes after ARHGAPIIB overexpression, in addition to having more upper cortical neurons.
Yeah, of course, Daniel is asking about folding. So not really, not very little actually. So the way I see it is that ARHGAPIIB, when you add it to the mouse, it increases folding. Mouse that is already lissencephalic starts getting folds. When you do the same in marmoset, that is also basically lissencephalic, it can also induce folding. And but where you add it to Ferret, that is at the beginning durencephalic, folding is not really increased, suggesting that perhaps in Ferret there is enough, or in durencephalic organisms in general, there is a certain plasticity in development that will allow them not to massively change the brain shape, but developed other abilities to pack these additional neurons. So what is very important in ARHGAPIIB ferrets compared to the control is that the density of neurons, particularly in these upper layers, was really increased and very, very significantly. And this is something that is also what we can see if we look at primates, for example. They will have an increase in density of cells between different primate species, more than the actual increase in folding. So ARHGAPIIB, in short, I would say as a hypothesis that in organisms that are lysencephalic, it can induce folding, but in organisms that are already durencephalic, it might have different effects, which is more increasing the density of neurons. And this it doesn't depend on ARHGAPIIB. This depends on the development of that organism in general.
Okay, then we have another question from Carla Perrone Capano. Again, many thanks for a beautiful talk. Do you have insights on how the morphoregulatory complex is modulated during development? Is it involved also in adult neural stem cells proliferation?
Yeah, this is a very interesting question, and we didn't look at that. It's since the similar cells should be involved, it's highly possible, but we haven't looked at that, and this could be, of course, one of the ways to target neural stem cells in adult. This is something, again, the problems are always the same, that in mouse the systems might be different, so we would have to go into another model organism for that.
Okay.
Rossella, I have a question, if I can.
Yeah, please.
There are no questions from YouTube, actually. Great talk, Nereo. Thank you. I like very much the different approach, the evolutionary approach, the pathological approach, and the development approach. Actually, I have some different questions, but one possibly most urgent is, what about the function of PALM-D? You're saying that it's a morphogen, right? And if understood correctly, the idea is that the only difference between mouse, let's say, and humans is the fact that there is a different localization inside the cell. So there is a new, a different localization under the membrane. So is it a gain of function, an acquisition of a new function, or is it losing its normal function, which allows to acquire new functions? So it's more on the function of the protein and what is changing in evolution.
Thank you for the question, and also for your very nice words. So yes, there are two isoforms that, as far as we know, exist in all mammals. And what I think is that the very important thing is which is going to be expressed in different cell types. So the one that is bound to the plasma membrane is expressed in the human brain, and this is the one that will be involved in process growth, and I will come back to the possible mechanism there. But in mouse, this isoform is not expressed in the brain at all, during the development at least, but it might be expressed in some other cell types. And indeed, when we express this isoform in various cells in culture, it will always induce processes, even in fibroblasts. So this might be its generic role. It's just that during the neocortex development, this becomes very relevant for in evolutionary terms. And the mechanism it does it is likely this, together with adducins, in anchoring actin cytoskeleton to the plasma membrane. So this can probably increase stability of the newly induced process. So when you bring cytoskeleton in, it probably becomes more stable, and then it remains there for longer. I actually, in support of this hypothesis, we noticed that when we overexpress PALM-D, that cells keep their processes throughout cell cycle, even in mitosis, which is something these cells usually don't do. They usually retract processes in mitosis and round up. But when PALM-D is so massively overexpressed, they will keep them, suggesting that probably stability of the process is increased.
And then we have the last question before moving to the next speaker, that is from Lucia Verrillo. And she's asking, do you have information about the potential involvement of ARHGAPIIB in brain cancer forms?
Yeah, so this is something that definitely Lebo Wilhelm-Huttner is working on the potential involvement in cancer or in any other neurological disorder. And being human-specific gene, we have this would be really interesting to find how it could be involved in some of the pathologies that we were very unlucky in modeling in animals so far. So this is something that has been working massively on, and a lot of clinical collaborators, I think, are taking part in trying to find these mutations in patients.
Thank you, Nereo. Thank you very much again.
Thank you very much. Thank you.
Okay, so I think we can move to the next speaker. Rossella, do you agree?
Yeah, please.
Before moving to the next speaker, I have a couple of technical points to raise. If you are interested in getting any attendance certificate, please write to Simona Palladino and ask for this kind of documents. The email was in the announced email, but I can write it back in the chat later on. And the second point, I want to invite, on behalf of Ennio Del Giudice, the president of Neapolitan Brain Group, I want to invite the scientific committee of NBG to stay after the meeting and have a small and a brief meeting to decide the next meetings for the group. So the scientific committee, please stay for some further minutes after the webinar. And with that, I want to move to the next speaker, who is Daniel Del Toro Ruiz. Daniel, please switch on your camera and your microphone. Daniel did his PhD in Barcelona on Huntington disease, and then he joined the lab of Rüdiger Klein at Max Planck Institute of Neurobiology in Munich, where he studied the role of FLRT proteins in cell migration during cortical development. His work has provided a molecular mechanism linking neuronal migration and cortex folding that led to the Young Scientist Award from Max Planck. Recently, he started his own lab at the Faculty of Medicine at University of Barcelona, where he continues to study how neuronal migration influences cortex morphology. And today, he will talk about synaptic proteins guide cortical migration and influence cortex folding, which perfectly fits our half-day meeting on evolution of brain cortex in mammals. So Daniel, please.
Yeah, so thank you so much for this nice introduction, and I'd like to thank the organizers for the chance to present our work in this nice webinar. So I'm going to share the PowerPoint, and the talk, as you said, is about the role of synaptic proteins. So now, where is the, I'm looking the PowerPoint, and why? Oh my, okay. Somehow, sorry, where is the, now I have issues here now, but 1 second.
Yeah, don't worry.
No, it should be fine. I don't know why. Now it's not detecting the PowerPoint. Straight. Now, here, sorry. So I hope that now you can see the PowerPoint. Is it fine?
We do, it's fine.
Yeah, so so thanks again for this nice, for the chance to present our work in this nice webinar. So the talk, as you can see here in the first slide in the introduction, was that mainly at the Max Planck Institute during my postdoctoral stage at the Ruby Kleins, and it's about the role of synaptic proteins that are also expressed during cortical development, but not for making synapses, but to control neuronal migration and how this can influence cortex folding. So as we have seen in the previous nice talk, brain size has changed dramatically across mammals, and this mostly accounts for an expansion of the cortex, which comes together with the formation of gyrus and sulcus, which are critical for fitting this large cortex into the kernel volume. So if we now focus on the cortical development of the disencephalic smooth cortex of the mouse, we find in the apical site, in the ventricular zone, the apical radial glial cell, which is the primary stem giving rise directly to neurons, but also indirectly through the generation of basal progenitors that are located in the subventricular zone. These neurons will migrate up to the cortical plate where they settle and then differentiate. In contrast, if we compare this with a genencephalic brain, what we find is a massive expansion of basal progenitors, as we have seen in our previous talk. And it's so big that the SVC, the subventricular zone, is further subdivided into outer and inner SVC. And within the outer subventricular zone, we find a new type of radial glial cell, which is called the basal radial glia, which is not attached to the apical site compared to the apical radial glial cells, but yet contributes to the divergence of radial fibers, which is important for the gerification of the brain, together this massive expansion of neurons due to the increased cell proliferation. Indeed, in the mouse, expanding the number of basal progenitors can lead to cortex folding, and this was shown nicely by Magdalena Gott's lab in 2015, where by in utero electroporation they were knocking down TRIM1, and this led to a massive expansion, as you can see here in the electroporated site, of TBR2 cells, which labels basal progenitors, compared to the contralateral site, as you can see here. And in the area of electroporation, they could see folding of the cortex, as you can see here by these two arrows, just very close to the area of electroporation. However, in the mouse, not always increasing the number of basal progenitors is enough to drive folding of the cortex, and this was nicely shown by the work of Caligari's lab in Dresden, where they use now here a virus. It's called 4D, which induces the expression of cyclin D1 and CDK4, and they could show that in the mouse, this virus is able to almost double the number of basal progenitors, here stained in white with TBR2, and this leads to a massive expansion of the cortex, as you can see here, compared to the control brain. Is larger, is also thicker, but yet it remains smooth. However, the same approach in a genencephalic brain, which is a folded brain, is able to increase the number of sulci and gyri present in this brain by default. And this was clearly showing that in genencephalic species, there are other mechanisms in addition to increased cell proliferation, which are important for folding of the brain, and that these mechanisms are not present in a mouse brain. Indeed, the current hypothesis states that cortex folding is due to a combination of two events. First, we need an amplification of these basal progenitors, of course, but also we need a divergent radial migration. So it's very important how these neurons will migrate. And this is where I am focusing here, how migration impacts on cortex folding. Indeed, the way neurons migrate is very different if we compare the mouse versus a genencephalic species. In the mouse, newborn neurons, they mostly will follow their related radial glial fiber up to the cortical plate, meaning there is very little lateral dispersion. And this was nicely shown using retroviruses to label a single radial glial cell, and they could follow the authors, just their progeny, all the newborn neurons of this radial glial cell, and they could observe that neurons mostly follow their related radial glial cell. So in the mouse, when neurons are born, will dictate their lateral position in the cortex. However, in the ferret, in a genencephalic brain, things are very different. Neurons, when they are born, are able to switch several radial glial fibers, and they can disperse in the lateral axis up to several millimeters. This was nicely shown by work of Víctor Borrell's lab in Alicante, Spain, where using a retrovirus, they were able to label a small area of basal progenitors, and they could follow their progeny, where the neurons will be located in the cortex, and they could observe that neurons that were born from this tiny area were able to populate a very wide area of the cortex. So these neurons were able to disperse laterally a lot. And this is only happening in the encephalic species, and the mechanism still remains largely unknown. Just to give you a hint of the relevance of cortex migration in folding in humans, several genes which are linked to abnormal cortex folding are indeed related to cortical migration instead of cell proliferation. Of the list that I'm showing here in this slide, the first two genes, lissencephaly 1 and double cortin, are related to cortical migration in mouse and ferret, and account for 75% of all cases of type 1 lissencephaly in humans, where the brain size remains unchanged. There is the same number of neurons, but yet the cortex remains smooth. And this shows how important is cortical migration for shaping the morphology of the cortex. So during my postdoc, and now here where I'm in Barcelona, I'm focusing on the study of a particular protein family called Ferrets, and they are single transmembrane receptors. And in their ectodomain, they have an important domain called the leucine-rich repeat, which has this banana shape. Okay. During development, all Ferrets, and there is Fer 1, 2, and 3, they are all expressed in the mouse cortex. Fer 1 is more widely expressed from the subventricular zone up to the cortical plate, only in neurons, even though it's expressed in the subventricular zone where basal progenitors are. Fer 3 is expressed from the intermediate zone up to the cortical plate, and we know that these two genes, Fer 1 and Fer 3, are also co-expressed in narrating neurons from the intermediate zone, and Fer 2 is just restricted to the cortical plate and is expressed only in neurons after they have migrated. So in 2014, together with Elena Serravaque, which now has her own lab in Oxford, and she's very good getting the crystal structure of several receptors, we got the structure between Ferrets and different ligands. And what we found is that through their lateral side here in blue, Ferrets interact with the most N-terminal domain of Ang5 receptors. And this interaction is repulsive. Ferrets repel Ang5 neurons. It's only unidirectional. And what we could observe is that this interaction is important to modulate how neurons migrate in the radial axis. So Ang5 neurons will slow down when they sense or interact with Ferrets. But at the same time, Ferrets, through their concave side here in orange, they are able to interact in an homophilic way. Ferrets interact with dense cells through this concave domain, and this interaction is adhesive. Ferret cells stick to each other. And this we could show that it's important to modulate how neurons disperse in the lateral axis. And this feature is quite unique because very few genes are able to do that. So it's nice to see that the same protein here, Ferrets, can achieve different effects just based on different interactions on different surfaces of the same ectodomain. So just to give you an example of the effect of lateral dispersion, here I'm just showing you a movie of an electroporated brain. In our previous talk, we have seen how this works. So we do this at E15, we make it transparent at E18, and you will see now when we go through the stack, all neurons that are labeled with GFP, just a reporter gene, they show this nice homogeneous salt and pepper distribution, as now you will see here in this magnification. And because these neurons are so homogeneous, the intensity fluctuation of the GFP is very small because we can see a GFP in all the regions. Now, if we do the same approach, but now we overexpress FLIR3, and we do the same, we do electroporation 3 days, we fix the brain, we make it transparent with cubic or iDisco, and then we ask where these cells are located, how is their distribution. If now we go through the stack, you will see that in contrast to GFP, when we overexpress FLIR3, the cells stand to form columns. They do clusters in a repetitive manner. It's quite regular, as you can see here, and because they form these clusters, these columns during migration, is why we have these peaks when we measure the fluctuation of the GFP. So because we have the crystal structure between flirts and different ligands, we can make mutants. We can add glycosylation sites to use sugars as a way to block the interaction between all these proteins. And we can ask which interaction is important for this effect, for these clusters that we see when we overexpress FLIR3. By doing so, we could see that if we block the interaction between ANG5 and FLIRT, the clusters are still present, meaning this interaction is not important for this clustering effect. But if we now block this concave domain, so flirts cannot bind in an homophilic way, we can see that now the cells distribute in a way that is very similar to our control GFP. So this homophilic binding, this adhesive binding, is important for the clustering effect that we see in vivo when we do the gain of function. But then why this is relevant, or why that's important? Well, if we knock down flirts, we see a similar effect. Also, neurons cluster in the mouse cortex. So in the first image, we can label FLIR3 in blue because we have the beta-galactosidase as a reporter gene. And as you can see in the cortex, from lateral to medial, the cells in the lower part of the cortical plate look very homogeneous, salt and pepper. Now, if we remove FLIR3, you can see how on the lateral side, they stand to form these columns, these clusters, as we can see here. And this effect is enhanced if we knock down on top FLIR1. As I said, FLIR1 and FLIR3 are co-expressed in migrating neurons. So we can see that now these clusters, these columns, extend from lateral to more medial portions of the cortex. And this we can quantify, right? So if we check the lateral part, we can see that both the double mutant and of course the single mutant for FLIR3 show clustering. But if we go more medial, only the double mutant is able to show this effect because the phenotype is increasing and extending to the medial part. And why this is relevant, why clustering of the cells in the mouse cortex could be important? Well, what we could observe is that given that now neurons that are migrating are only going through discrete columns, as I'm indicating here, they arrive to the upper part of the cortical plate in a very heterogeneous way. Now we have these clusters on top of these columns because this is the way where they are entering into the cortical plate. And this makes the upper cortical plate to look very wavy because now it's heterogeneous, it's not homogeneous anymore as happens in the mouse cortex. And at the same time, we could observe this clustering and waviness in the upper part of the cortical plate. We could see sulcus formation in the mouse cortex, not in all the mutants, in around 30% of double mutants. And if we ask where do we see these clusters, this sulcus as early as E15.5, we could see in green that the sulcus formation was happening in areas that were close or within, in red, the areas where neurons cluster, indicating that the clustering of cells and the sulcus formation are related. There's a link between these two different phenotypes. At E17.5, 2 days later, this mild phenotype, as we can see here, of this sulcus formation looks better. We have examples that looks more mild, as we can see here, or a stronger phenotype in the image down below, where the marginal zone, as you can see here, is also entering. And here we have two sulcus forming. And these strong cases, this strong phenotype, are the ones that we can already visualize after dissecting the brain and careful removal of the meninges. We could observe this sulcus formation. And at this point, we were impressed because this was totally unexpected. And we were wondering what could be the mechanism behind this observation. Why do we see this sulcus formation when we remove the flirts? And the first thing we check, and also based on the previous talk and what I said in the introduction, was to check if there was an increase in cell proliferation. And to our surprise, when we check for markers of apical progenitors such as PAC6 or basal progenitors such as TBR2, or dividing radial glial cells with phosphobimentin, we could not observe major changes in the numbers of cells in our mutants compared to control brains. We only observe a 20% change, which is small, but yet it was significant in the number of phosphohistone 3 cells that labels dividing cells. But this small change, this 20%, we don't believe that explains totally the phenotype that we have. So we moved to check whether the integrity of the tissue was affected. This could be also another reason. So as you can see here in this image, the basal membrane is totally intact, despite this waviness formation of the cortical plate underneath. We can see the caja-2 cells, which are nicely underneath the basal membrane, as it should be. Even in the more extreme cases, as you can see here, where the marginal zone is going inside, the caja-2 cells follow, as happens in denscephalic species. And if we label the basal radial glia fibers, they don't detach. They just converge in the areas where sulcus form, as we are used to see in the ferret or in human brains, where the radial glia fibers converge in the areas where sulcus form. So because all these features look normal, we focus on the distribution of neurons, which was the last thing to check. And as you can see here, I present you on top mild case, and on the bottom, strongest one. We first check for the layering of the neurons. So we label with Cook's one the upper neurons, and as you can see, they are in the upper side of the cortical plate in both mild and strong phenotypes. As you can see here, they are in the upper part. And if we stain with TBR1 the lower cortical plate neurons, they also occupy the lower part of the cortical plate. So the layering is totally normal. But as you can see, the proportion of cells is not. For instance, in the areas where sulcus are very deep, as here in the image down below, you can see that there are fewer lower neurons compared to adjacent parts, because it's getting thinner, the cortical plate in this area. But what happens to the upper cortical neurons? Here I'm just showing you an inset of an area with a sulcus and an adjacent or control section. And as you can see here, if we measure the total number of Cook's-1 cells that occupy the entire cortical plate that I'm indicating here in red, and as I show you here in this image, if we count the total number of Cook's-1 cells of upper neurons, we don't see changes. And this goes in line with the idea that we don't have an increased proliferation in these brains. But if we count how many have arrived to the upper part of the cortical plate here with this yellow outline, respect those that are still migrating, and we check that in the areas where sulcus form, we see that there is an increased number of upper cortical neurons that are already in the upper part of the cortical plate in the areas where sulcus form. And at this point, we had two options. It could be because, of course, the radial distance now is shorter, and that's why they arrive earlier and faster, but also it could be that these neurons migrate faster. So one way to address that was to do time-lapse imaging. So we can do electroporation. We can label this with a reporter genes such as mCherry or GFP, and we can track the cells in control and mutant brains. And because we track the cells, we can color code their paths based on the distance, based on their speed, in tracks of 20 minutes. And as you can see here, I'm just showing you the entire the raw data where every line is one cell. And as you can see in both data sets, control or in the double mutant, the range of speeds looks the same, right? In both cases, we see cells that pause for a while, so they move at 0 microns per hour. And in some moments, these cells are able to acquire and go very fast, above or 60 microns per hour during 20 minutes. So the range of speeds is the same in both data sets. But now, if we only plot the high-speed tracks, those above 60 microns per hour, we can see that they appear more often in the double mutant data set, meaning that the mutant cells are able to sustain for longer or to acquire more often these high-speed tracks. And this we can measure if we do the frequency plot distribution. You can see that in mutant cells is much more common to observe and much more frequent to see these high-speed tracks. So overall, mutant cells in the absence of flirts, not only they do these clusters, they also migrate faster. And with that, we just create this model. In a control brain, neurons have this salt and pepper homogeneous distribution. They also migrate in a coordinated way, and this helps to build smooth layers. Now, in our mutant brains, in the absence of FLIR1 and FLIR3, these cells form these columns, they form these clusters, and they also migrate faster. So we have this now heterogeneity of cells in the upper part of the cortical plate, and this creates tension, and one way to release this tension is by creating a sulcus. At this point, we were struggling because we couldn't understand why, in the absence of flirts, we have this adhesive feature, cells were clustering. This seems to be a little bit counterintuitive, right? We remove a protein involved in addition, and we cells clustering. Which was even more strange for us was that if we take this place out of context, control cells that express flirts with in vivo, they show this homogeneous pattern and salt and pepper. In vitro, on a cover slip where they are free to acquire any configuration, they form clusters, as we can see here, because flirt cells have this adhesiveness property, and cells are very sticky between them. But our mutant neurons, when they are on a cover slip, they show a random distribution because they lack this adhesiveness. And this shows that the clustering effect that we observe in vivo is non-cell autonomous. There has to be other proteins and that could interact with flirts that are important for the segregation of these two different cells, the cells that don't express flirts and those that were expressing. So to check what is the relevance of this phenotype during evolution, we team up with Víctor Borrell's lab in Alicante. He works with the Ferret, and he's an expert with this model. And this is really nice because when they are born, as you can see in this image, their brain is fairly smooth. And during the next 2 weeks, it gets folded because there's still a lot of migration going on. And in this folded brain, there are some gyroses, such as the spinal gyrus, and some sulci, such as the lateral sulcus, which are very conserved between individuals. And because of that, it is possible to check these areas earlier at P2 when still there is no folding, and we can see whether there is a difference in the expression in the mRNA in an area that later on will develop as the lateral sulcus, compare an area that later on will develop a gyri. And that's what we did. So here I show you the in situ, the expression pattern for flirts. And as you can see here in the ferret for FLIR1, areas that will develop a sulcus, in this case the lateral sulcus, you can see that the expression of a FLIR1 is lower in both the intermediate zone and in the OSBC, as we can see here in these graphs, is much lower compared to areas that will develop a gyrus. And the same applies for FLIR3. In the areas that will develop a sulcus, the expression levels are lower, as we can see here in these graphs, compared to an area that will develop a gyrus. And this is sustained at P0 and also at P6. And that's really nice because these areas that will develop a sulcus, which have lower levels of flirts, somehow resembles the phenotype we have in our mouse mutants, where we have lower levels of flirts and we get sulcus formation and not a gyrus. So what happens with humans? So what we did was to check the level of expression of a mouse at E14 in the lateral and middle cortex, and we compared with several cortical areas from E13, from 13 weeks post-conception human embryos. And as you can see, no matter which cortical area you take in humans, the levels are way lower compared to the mouse for both FLIR1 and FLIR3. And not only if we check different cortical areas, also if we check different cortical layers. So we can see here that if we check the cortical plate in the mouse, the levels here with the black whiskers are higher compared to human. Also in the ASBC, in the OSBC, where neurons will be generated, humans are almost 0. for FLIR1, and the same applies for FLIR3. We can see that the mouse is always higher, much higher than humans in all these areas. And it's not only if we check for different cortical layers, also if we check for cell types. Here, if we just compare neurons from the mouse, we can see that the levels of FLIR1 are way higher compared to humans. And also for FLIR3, neurons express high levels compared to humans. So we can see that during development, we are literally natural knockout for FLIR1 and FLIR3. So after this study was published, where we could observe these strong effects of FLIR's knockdown in cortex folding, we were wondering if other proteins could be important for this phenotype, because as I show you, we see that this effect is non-cell autonomous. So we focus on latrophilins, because in 2012, the lab of Andy van Gogh showed that FLIRs are endogenous ligands for latrophilins, that these receptors are the receptor of the black widow of latotoxin, the venom of the black widow spider, and it was orphaned for several years, for several decades. And Andy van Gogh could show that FLIR's binding latrophilin is important for synapse formation and maintenance. And recently, 1 year ago, the lab of Thomas Sudhoff showed that in the hippocampus, FLIRs in axons from C3 and from the entorhinal cortex are co-expressed with tenurins, and these two proteins bind latrophilins. Latrophilins are present in the dendrite, and the binding is in trans, and this interaction of all these three proteins together is important for the formation and maintenance of excitatory synapses. So we wonder whether latrophilin and also tenurins, which are important for synapse formation, are also expressed during cortical development, and whether they also participate together with FLIRs in this process. So what we did was to team up with Elena Cheradakis' lab, and we got the crystal structures between FLIRs and latrophilin, and it was surprising to see that the same concave site that FLIRs use to interact between themselves is the same surface that they use to interact with latrophilins, but only through one particular ectodomain called the olfactometin domain. And latrophilins, in turn, through their lectin domain, are able to interact with tenurins to a quite large area of their Whitey shell domain. So all these three proteins can interact and form a triple complex. So when we check their expression during development, we could find that all latrophilins and all tenurins are expressed during cortical development from E13 onwards. And here I'm just showing you the in situ for latrophilin 1 as an example, but also applies for latrophilin 2. So as you can see here, with in situ is highly expressed in layers that are enriched for neurons, such as the cortical plate and the intermediate zone, but also it's highly expressed in the apical progenitor side, where radical glia cells are located, and here we label them with phosphoaugmenting antibody, and we can see the mRNA for both latrophilin 1 and latrophilin 2. In contrast, tenurins, here I'm showing you tenurin 2, but also applies for the other 4 tenurins. They are mainly expressed in neuron-enriched layers, in cortical plate and intermediate zone, and there is almost no expression in radical glial cells. As we can see here, just to show you, this is the mRNA for tenurin 2, also for tenurin 3, and they are expressed in cells that are expressing CD2, which is a marker of lower cortical plate neurons. So just to confirm that all these three proteins could interact and form a complex, we did an in vitro assay. We basically cultured embryonic cortical neurons from E13.5, and we do the staining with antibodies for surface FLIR3 and surface tenurin 2, because these two proteins are co-expressed in narrating neurons. And we added into the media latrophilin 1, the leg-off domain, which means that it can bind both tenurins and FLIRs. And we could find events where all these three proteins were colocalizing in both neurites and in the cell body of these neurons. And we could confirm that this interaction was indeed happening doing super resolution, as you can see in the image down below, because of course with confocal sometimes it's difficult to know whether three proteins are truly colocalizing. And here we could find events where all these three were as close as 30 nanometers. And there were also other events here with this gray arrowhead where all these 3 proteins were very close. So this shows that in this triple complex can form in vitro. We also have evidence for in vivo. So this was our proposed model. So basically, migrating neurons that express tenurins and flirts in cis, they interact with latrophilin in trans, and latrophilin is present in radial glia fibers because we have seen that latrophilin 1 also 2 are expressed in the radial glial cells in contrast to tenurins. Just to focus on this interaction between neurons and radial glial cells, we used nanofibers to mimic the radial glial cell fibers, so it's easier to work with. And this system is really nice because we can coat nanofibers with any protein. We can culture on top cortical explants. We see how these neurons exit the explant and how they migrate along these fibers. It's also nice that we can electroporate the explants, and we can see, as you will see in this movie, how a neuron is exiting, and at the beginning it has this nice multipolar shape. The time is in hours, and at some point is shifting to this bipolar shape and moving along a nanofiber in a very similar way that we could expect from in vivo experiments. We can fix them and stain them, and as you can see here, we have 2 neurons that are moving along these fibers, and they have this nice leading process that happens in vivo and a short trail, and you can see here how this leading process is able to switch to a neighboring fiber, which also occurs in the mouse brain. So we did this assay where we cultured these cortical explants, and what we did was to coat them with latrophilin wild type or a mutant that no longer binds flirts or tenurins. And as you can see here, after 2 days in culture, we measured the distance of the cells after they were migrating. And if we coat with latrophilin 1, the distance is shorter. These cells migrate less compared to our control situation. That's why the colors are more goldish. So if cells migrate less, we color code them with this more bluish or magenta color. If cells migrate more, we color code them with more yellowish, red, or green color. And as you can see here with the latrophilin 1 double mutant, we have a nice partial rescue. As you can see here in this quantification, respect control or single mutants for latrophilin 1. What was surprising is that the same explants where we can see these cells migrating, we can also analyze axonal extension because cortical neurons also extend their axons while they are migrating. And we could not detect differences. So axons, which also express latrophilin, which also express tenurins and flirts, don't show any response and extend as well in nanofibers coated with latrophilin 1 or the double mutant compared to our control. So the effect of latrophilin is only happening at the level of the cell bodies. And this we know is because of a repulsive effect. And one way to observe that is to do the stripe assay. So here I'm just going to show you a movie where we can make on a dish a pattern with proteins. Here in the red stripe, we just coat with latrophilin 1. And in the green stripe, here we have the latrophilin 1 double mutant. Here we have a cortical explant where we have electroporated with a GFP to label cells that express flirts and tenurins. And as you can see in this movie, the majority of these cells, when they touch the red stripes, they just show this repulsive event. The neurites retract, and only there was one neuron that was very brave, this one, to go through the red stripe. So there were a lot of repulsive events. So at the moment, the neurites that express flirt and tenurin touch the latrophilin 1 protein, they show this repulsion. And this effect that happens at the cell body and neurites is not happening with the axons. So here we have the similar explant where we can see the axons growing. As you can see here and here, the axons are growing, and you can see that they go straight, and they don't care whether there is a stripe with latrophilin 1 or the double mutant. So this effect is only happening at the level of the cell bodies, which is very interesting because flirts and tenurins are in both regions, but only neurons and cell bodies show a response with latrophilin 1. So just to give you a hint about the relevance in vivo, we did the gain of function experiment where here we just labeled the latrophilin 1, tenurin 2 with the HA. We overexpress it, and we could see that when we overexpress tenurin 2, the neurons tend to accumulate in the intermediate zone, and very few are able to go to the upper part of the cortical plate compared to our control or with our mutant tenurin 2 that is no longer able to bind latrophilin, showing the specificity of this effect. And just to show that this is because neurons are slower, here I am showing you a time-lapse movie where we co-electroporate tenurin 2 in green and M-cherry in red to also label control neurons that are not expressing tenurin 2. And as you will see in the movies, neurons that express tenurin 2 here with these red tracks are slower and remain longer in the intermediate zone compared to control cells that go up to the cortical plate. And this effect is nicely rescued or almost rescued if we use a tenurin 2 mutant that is not binding latrophilin. So in this movie, you will see if we overexpress tenurin 2 and we follow with the green tracks the neurons, they mainly remain in the intermediate zone, while in red ones, our control neurons were able to go up to the cortical plate, so they are faster. And if we now overexpress a tenurin 2 mutant that is not able to bind latrophilin, we rescue this effect. Finally, just before going to the acknowledgements, one experiment to show the relevance of the interaction in vivo, if we disrupt the endogenous interaction between latrophilin, flirts, and tenurins by expressing a secreted form of latrophilin 1 that is going to compete and disrupt the endogenous interactions, we also see a strong delay in the migration. Fewer cells go to the upper part of the cortical plate compared to our control and compared to our double mutant latrophilin 1 that is not able to interact with flirts and with tenurins. So as a summary, this is the slide. So I hope I could show you how nice it is to have the structures of all these proteins, because then we can use mutants that specifically block all these interactions and allow us to understand what is the function and in which process they are participating in vivo. So the take-home messages are these ones. So if we remove FLIR1 and FLIR3 from the mouse neocortex, this can induce sulcus formation. And it's not because we have more cells, but just because they now migrate in a different way. They make these clusters, they are now heterogeneous, and they also migrate faster. And we also propose that the expression of flirts inversely correlate with folding across species. So the lower the flirts, the higher chance to have a folded brain, as happens with ferret and as happens with humans. And finally, I've showed you, and that's a recent study that we has published this year, that during development, flirts are interacting with latrophilins in trans together with tenurins, and this interaction is important not for making us synapse, but for controlling how neurons migrate in the radial axis. So I'd like to thank a lot of people, of course, Rude Klein, because all this work was done where I was there as a postdoc. Tobias, PhD student that now is doing his postdoc at ETH Zurich for all the time-lapse imaging. Eric, former postdoc that did all the analysis with human and mouse expression for flirts. Gonul for technical assistance, and Sarah, postdoc in the lab that did all the time-lapse movies with the gain of function for tenurin and also for latrophilin. Our collaborators, Elena, which has her own lab at Oxford, and she's awesome for making all these crystal structures and all these mutants and all her lab. Vitor Borrell for the awesome work with the ferret, and Magdalena Watts for nice discussions. And as said during the introduction, now I'm in Barcelona. I'm running the show with the flirts here at the university and the Faculty of Medicine, and I'd like to thank you all for your attention. Thank you.
Great. Beautiful talk also by Daniel. Any question from the audience? I'm checking the chats. I have a naive question in the meanwhile that people think about questions and write down in the chat. Daniel,
yes.
I was wondering. I'm C. elegans guy, so I'm not really inside the mouse work. But I was wondering whether you can test any phenotype, behavioral phenotype in your mice altered in FLIRT expression in terms of, I mean, possibly the double knockout is too heavy, the manipulation, but I don't know if there is any conditional or any overexpression model where you can assess if your increasing folding might have an impact on behavior.
Yes, so that's a really good question. Indeed, basically in all the studies working in the mouse or other systems, usually we focus during development and we don't check what happens postnatally. And I always get the same question as, okay, are your, the mice with a sulcus and folding are more intelligent? So that's something that we want to address now, just to do some behavior. We plan to do MRI to check before doing behavior which areas of the cortex have this folding and make the behavior accordingly to the area. One thing I can tell you about these mice, because they are viable, is that at least for their social interaction is increased. This we have seen. So at the moment that you add a new animal, they are very curious and they spend longer time compared to control mice. And something that I find very interesting, which I don't know whether it's a sign of being more intelligent, is that it seems they are sexually more active. So I don't know. That's something that I've observed with these mice, and so it could be. And I hope that these effects translate to some behavior. At least in humans, I have to say that having low, so fewer folds or more, in case of the polymicrogyria, both have side effects. So in case of humans, having too little or too more is deleterious. We need to see what happens in the mouse.
Thank you.
Thanks, Daniel. I also have another naive question. So related to the proliferation, because you said there is a 20% increase
Yes.
of mitotic cells.
Mm-hmm.
How does it correlate with this, or if it does at all, correlate with this multi-complex of protein?
Yeah, so basically we don't know. So FLIRTs are not expressed. So that's the interesting part. So FLIRTs
is, right?
Yeah, so FLIRTs are not expressed in progenitors. This we have checked for FLIRT1, for FLIRT3, and also for FLIRT2. So we were surprised to see that there was an effect in the number of dividing cells. It's small, but yet is there. So regarding laterophilins, they are expressed in progenitors. So we cannot discard that there is a non-cell autonomous effect going through the laterophilin signaling. This could be something to check. So far, there are knockouts for the laterophilins, but all the studies have been focused postnatally, because laterophilin has been always linked to synapse formation. So it will be interesting to see if in laterophilin knockout brains, there are some effects at the level of cell proliferation that could help us to understand why, in the absence of FLIRTs, there was this small but yet significant effect. So far from the FLIRT-dependent signaling, I cannot imagine any way that could explain that. So I will assume that it's a non-cell autonomous effect. But I think that even though it is there, I think that the effect on cortex folding is not that much, not only because it's just only 20%, but also because it happens in all mutants, right? And sulcus formation is not happening in all mutants. It's just 30% of them. So indeed, the mouse cortex is very difficult to fold or to induce folding. There are a lot of mechanisms that try to compensate and try to prevent this from happening. So that's why I think that the way neurons migrate and the fact that at E15.5 the sulcus appear always very close to the areas where cell clusters, there has to be a strong correlation between these two different phenotypes.
Thanks. Great data.
Thank you.
I don't see other questions. Rosella,
Yeah, there is one.
I can't see it.
Yeah, 1 second. There is one. I saw one raised hand by Nereo.
Yes, hi. Hi, Daniel. I was wondering if you are thinking of manipulating flirts in Ferret or any other gyroencephalic model organism.
That's a really good question, and also based on a paper that Kawasaki published, few years ago, where he knocked down CDK5 in ferret in neurons, and they were moving slower, and he could observe that the sulcus are also slower. So there's an effect in cortex folding is less folded if neurons migrate less. So that is something that could be very interesting to check, and also with organoids. I would be really curious to see if human organoids, if we increase the levels of flirts that naturally are not there, if somehow affects the way the neurons migrate, if they will show clustering, or at least if they will move maybe slower. That's something that I'm really interested to check. So, and I'm really open to just to collaborate with anyone. So yeah, I'm totally free, and I'm happy because my expertise is not with ferret or even with organoids, although I like to step in and to learn and apply at least organoids to the work that I'm doing.
Great, let's be in touch.
Yeah, sure, thank you. Yeah, it would be nice.
If there are no other questions, I have one. Rosanna Del Gaudio? No.
Maybe she used the wrong chat.
Okay, so Daniel, a couple of quick questions. Can you exclude any role of natrins? Because if I remember correctly, ANK5 is a natrin receptor, and hence the name. You know that ANK5 is the name.
Yes.
From C. elegans. And the other question is, any idea, possibly you said that, but I missed, any idea on why flirt is downregulated in human brain, if I understood correctly?
Yes. Well, sure. So very good question. So regarding the first one and the role of natrin, yeah, so there was a publication about the role of ANK5B, important for the transition from multipolar to bipolar state of migrating neurons to the intermediate zone. And even though ANK5s are expressed there, so far natrin during cortex development, the levels are extremely low. So I don't think that natrin by itself will be important, even though DCC and ANK5s are expressed in migrating neurons, and that's why maybe the role of natrin is used or is just taken by the flirts, because are also ligands of ANK5s. Regarding the last question, so why in case of humans the levels are lower, so we don't know. Also in the case of ferrets, why in areas that will become a sulcus are also lower, we don't know. In ferret, and also work from Vito Borré's lab, it is clear that there is a genetic protomap in the cortex that is not homogeneous, where some areas have increased cell proliferation, and the genetic program is set before that. So it could be that from other transcription factors regulators, flirts can be regulated, and this could explain why some areas have high and low expression, and why in humans overall the levels are so low. The reason that I could think of is that for neurons to migrate faster and to have increased freedom is better if they are less sticky with themselves. So the way I see the neurons migrating in the mouse is like people running while holding their hands. So it's a nice way to migrate in a synchronized way. But it is better if neurons have more freedom to acquire different migration speed and be able to move laterally a lot. So maybe this is one way to remove stickiness, to make the tissue softer. Also, a nice paper from Wieland Hunder's lab showed that sulcus areas in human sections are softer compared to areas which will become agile. So I think that the stiffness of the tissue is important, and maybe reducing these proteins helps to acquire a complex morphology of the cortex.
Great, thank you very much.
Thank you.
Rossella, I think.
Yes. Okay, thank you, Daniel, again. And now we switch to the third speaker, Cristina. Can you turn your video on, Cri? Okay. So Cristina did her PhD in basic medical sciences at the School of Medicine at the University of Patras in Greece, in the lab of Stavros Taraviras, where she focused on mechanisms regulating fat commitment, decision of embryonic and adult neural stem cells. For her postdoc, she moved to the Max Planck Institute of Psychiatry in Munich in the lab of Silvia Cappello, and there she focused on the mechanisms regulating human neural progenitor function in normal brain development, in evolution and disease. From February 2021, she will start a new position as assistant professor at the Medical School of University of Athens in Greece, where she will continue modeling human brain development and disease like cortical malformation and ciliopathies. And we wish you good luck for that, for the new career. So today she will talk about extrinsic function of ARHGAPIIB in neuronal progenitors during human brain development. Thanks, Christina.
Okay, thanks a lot, Rossella, and also thanks to all the Neapolitan Brain Group for this invitation. I will try to share my presentation. So I hope you see it now and the mouse.
Yes, yes.
Okay, so as Rossella said, I will talk to you about extrinsic mechanisms that regulate neural progenitor cells during human brain development. So as we already heard from Daniel and Nereo, the developing of the cortex is a highly orchestrated process, and it's quite similar in several aspects during development from mouse to human. So as a small recap of what we heard already, during brain development, the key neural progenitor population that occupies the developing cortex is this population, which are called apical radial glial cells. They occupy the ventricular zone, and they keep the attachment with an apical process to the ventricular surface of the developing cortex. They also have a basal process which span the whole entire cortex to the PL surface. They are important for proliferation and expanding their numbers at early stages during development, and later on they can give rise to a second progenitor population, which is called basal progenitors or intermediate progenitors, that occupy a second layer, the subventricular zone. So then both of these cell types can differentiate into neurons that will migrate and will form the cortical plate of these 6 layers of the cortex that we found in the adult brain. So although the key steps are quite similar between mouse or human, or lysencephalic and gyrified species, we see that in gyrified species, and in particular in humans, we have an increased number of neurons that are found within the developing cortex, and the generation of this SURC gyri that we already heard from Daniel. And it is believed that one of the key mechanisms or key factors that lead to this neuronal expansion in gyrified species and in humans is the existence of a unique progenitor population, which is called basal radial glial cells, which are very rare during mouse development or in other lysencephalic species. So this population is formed another layer, which is called outer subventricular zone, and it is very similar with the apical radial glial cells, with the only difference that they lack this attachment to the apical side of the ventricular zone. So as you can understand, the tight regulation of the mechanisms that regulated both neural progenitor population and differentiation and neuronal migration at the end is important for the proper cortical development, while changes in these processes or cellular molecular mechanisms can lead to cortical malformations or other neuronal disorders, such as neuronal migration disorders. And we have, for example, in this scheme, specific types of neuronal migration disorders that can be observed in humans, such as lysencephaly type 1, which is characterized by a smoother brain, with a simplified version of SURC and gyri. We have the subcortical band heterotopia, which is characterized by a second layer of gray matter within the cortical area, while we also have other disorders which are characterized by the fact that specific amount of neurons fail to go to their correct place, but they instead located in different positions. And in particular, in periventricular heterotopia, we find clusters of neurons that are found exactly next to the region of their generation, meaning the ventricular zone, failing to migrate to the cortical plate. While in lysencephaly type 2, these neurons cannot stop into the correct position, but they continue migrate to exceed the PL surface of the cortex. So the past few years, a lot of effort has been done in order to see both the cellular and the molecular mechanisms that involved in such disorders. And although they are called neuronal migration disorders, it is known that also the morphology of the progenitor cells is also very important in this formation of the brain malformations. And to do so, we and others, we are using model systems, of course, to study this such processes. And we use in vivo animal model system, the mouse model that it was so nicely introduced already from the two previous speakers, and the ferret model, which is a gyrified species that Nereo said. And also very recently, it's also the use of other primates such as macaque is also used as a more similar to the human cortex. Although all these models are non-human specific, obviously, so they lack human-specific features that can contribute to diseases that are found in individuals. And actually, this is the reason why some of these diseases cannot fully recapitulate it in these animal models. So for this reason, we use in vitro human-specific model systems, which are the 2D culture of neural progenitors and neurons, but also a more complex 3D structure, 3D cultures, which are the steroids, simplified 3D structures which have neurons and progenitor cells, and also the most organized and more highly complex cultures, which are called cerebral organoids, that can recapitulate the developing human cortex, as I will talk to you in the next slides. And finally, we can also drive these organoids to generate specific parts of the brain and then to fuse them together in order to have a more complex system to answer key questions regarding connectivity or interactions between different brain regions. And also, again, under a human-specific system. So the original protocols that have been used for cerebral organoids are based on intrinsic mechanisms that these cells have upon just culturing in normal neural differentiation medium to generate neural progenitors and neurons. And actually, we can start from induced pluripotent stem cell lines, so human-specific lines, which can be generated can be reprogrammed, generated from fibroblasts or PBMCs or other cell types that can be isolated from individuals, control or patient individuals. And as soon as you have an established iPSC colony, you can make these colonies into single cells. We plate the cells in low adherent conditions in order to form these embryoid bodies, which is again an intrinsic characteristic of these cells. And then just by culturing these embryoid bodies under neural induction medium, we have the formation of the neuroectoderm. And then by applying neural differentiation medium and by just giving structural scaffolds such as matrix gel, we have the generation of these cerebral organoid structures, which have within these 3D structures ventricular-like areas that resemble the ventricles of the developing brain, which then are surrounded from progenitor cells and neurons. So it has been shown in the past few years that this cerebral organoid culture can recapitulate nicely the early steps of human brain development, and in particular the stages that are found at the end of the first trimester and beginning of the second trimester. However, more recently, several protocols have been generated, and they can actually keep these organoids up to 1 year or even more in culture in order to give the time to these cells to mature properly, and in order to study neuronal maturation, migration, or connectivity in a human-specific system. But how can we? How do we know that the cerebral organoids recapitulate this human brain development in vitro? So if we take a section from a cerebral organoid and we stain with markers that we know that are expressed specifically in different cell types that we found in the human brain development, we see that around these ventricular-like structures that we have in the cerebral organoids, we have cells that express markers such as SOX2, PAC6, or nestin, which are expressing in the radial glial cells in the developing brain in vivo. In more basal locations, we also have basal progenitor cells, TBR2 positive or HOPX positive cells, while in the cortical regions of these organoids, we have neurons that are also distinguished by their time of generation by expressing several markers such as TBR1 or COX2, which are markers from neurons that are generated early on during development, occupying the deep layers of the cortex, or neurons that express SATB2, which is an upper layer neuron marker, meaning the neurons that are generated later on during development. But the most important thing of these cerebral organoids that makes them more useful than the mouse model, which is the most commonly used, is the existence of these basal radial glial cells, these human-specific or in general gyrified-specific progenitor cells, which expressed several basal radial markers such as HOPX, and they have also this basal radial specific morphology with the basal process and the lack of the apical attachment. So using these cerebral organoids, we can perform actually most of, if all, the techniques that we can apply also in the animal models. So, for example, we can perform immunohistochemistry or in situ hybridization techniques in order to see the set of architecture of the progenitor cells and the neurons within these cerebral organoids. We can perform FACS analysis to measure the ratio between the different cell types, or bulk and single cell RNA sequencing experiments in order to identify the molecular identity of these cells within the organoids. But also we can perform more functional experiments such as live imaging, where we can label these cells in vivo with a GFP protein and then monitor them under the microscope while these cells are still alive. Or we can perform electrophysiology experiments to measure the neuronal activity specifically on a human context. So using these cerebral organoids, we can now answer key questions regarding normal brain development and evolution, or we can model brain-related disorders such as psychiatric disorders and developmental disorders. And also we can use these organoids for drug testing and viruses, like the Zika virus that Tereo also said before, that it is known to cause developmental disorders such as microcephaly. And also lately, the past few months, we have seen papers that were used that use organoids in order to study how the SARS-CoV-2 can infect actually brain cells. So in the lab, with the collaborators, we have actually tried to unravel such a mechanism of normal brain development. So in the evolution of the cortical neurogenesis from amniotes to the humans. We can also model psychiatric diseases such as autism or prenatal stress, and to show how stress can affect brain development. And also we are focusing on the developmental disorders such as microcephaly, lissencephaly, or heterotopias, that it is actually the main focus of the lab. But today I will talk to you about how we can use these organoids in order to model human brain development and psychiatric and developmental disorders by focusing on specific human-specific or at least human-enriched genes, and how we can investigate cell non-autonomous phenotypes during cortical development. So basically, work has been done already in order to identify human-specific features that act during brain development, and many studies have focused on identifying the molecular identity of these basal radial cells. And now we know that these cells are expressed some specific markers such as HOPX, DNC, BTPRZ1, etc. Today I will focus on one specific gene, which is called L-galase3BP, and which is an extracellular matrix protein. It is secreted from the cells, and actually we were focusing on that because we have seen that from published data that during mouse brain development, L-galase3BP is completely undetectable during brain development. Within the Ferret system, since we know, as Daniel said, that where the future gyri and the future sulci will be generated, we can see that in the areas that will be the future gyri, L-galase3BP is very much enriched in the progenitor population, comparing to the region, to the progenitors in the region where it will, where the future sulcus will be generated. While when we go to the human system, we see that the mRNA of alkali 3BP is very much increased in the progenitor population, in the rather glial population here, and in single cell sequencing data from from human developing brain. Alkali 3BP characterizes this population, this rather glial population, and its expression is declined upon neuronal differentiation. But also from the evolutionary perspective, the expression of alkali 3BP is also very interesting. So if we look at single cell data that RNA-seq data that were produced from cerebral organoids that were generated from cells coming from humans, chimp, or macaque, we see that the alkali 3BP is almost not detected in macaque brain cells. It is a little bit more increased upon evolution, more evolutionary close to the human species, such as the chimp. But actually, the alkali 3BP is very much increased in expression during human brain cells. So using this as a baseline, we have focusing on this alkali 3BP. We first of all validated in our hands that in the cerebral organoids that we generated in the lab, alkali 3BP is expressed in the rather glial population from single cell sequencing that we performed from control organoids, while its expression is declined upon neuronal differentiation. We also performed a time course analysis of the alkali 3BP expression throughout time in cerebral organoids, and we saw that the peak of expression of alkali 3BP is quite early during development. It is followed by PAC6 expression, a marker of rather glial cells, and it is before the peak of expression of basal located progenitors such as the HOPX positive and the OMS positive progenitor cells. So then we wanted to see how the protein level is expressed, which is actually nothing was known already about the protein level in the alkali 3BP in the developing brain. We first validated that the expression of alkali 3BP in the developing mouse brain is not detected. And then we went to sections from human fetal cortices where we saw that the alkali 3BP is expressed from early stages of brain development at gestational week 14. And it is also expressed at later time points in gestational week 20. And interestingly, what we see is that alkali 3BP is expressed high in the apical progenitor cells, but also in the progenitor population that it occupies the outer subventricular zone. What we also observed is that the expression of alkali 3BP has this puncta, so it is this puncta suggesting, and it is actually expected because alkali 3BP is a secreted protein, and we saw that these puncta are closely associated with the SOX2 positive progenitor cells within this region, and not with the NUN positive neuron cells, again validating its expression in the progenitor population. So having this as a start, we wanted to see how alkali 3BP can regulate neural progenitor cells and can influence human cortical development. And also because we have found 3 individuals that have cortical malformations such as microcephaly or periventricular heterotopia, and that and these individuals have point mutation within this coding region of alkali 3BP gene. And actually, all these 3 mutations were found in the exon 5 of the gene, which is also the most intolerant for mutations, suggesting that these mutations may be relevant for this cortical malformation that the individuals have. So we were able to obtain MRI data from the first 2 individuals, individual 1 and 2, that they have periventricular heterotopia and microcephaly, and we have performed a morphometric analysis on the MRIs of these patients, and we saw that there are changes in the cortical thickness within the cortex of these individuals in several positions, and actually also local gerifications alterations. And since these individuals, they only have this point mutation, they suggest that these mutations are causative for this malformation that the individual have. So trying to model how alkali 3BP could regulate human brain development and cortical malformations, we focused on the first individual, which have periventricular heterotopia. And by using the CRISPR-Cas genome editing system, we have generated induced pluripotent stem cell lines that have the point mutation that is found in individual 1 in heterozygosity in order to mimic exactly the situation that the individual exhibits. And then in order to see also collectively all the point mutations that are found in this region, we generated a knockout of the exon 5 of this gene, an IPS line that has this knockout of the exon 5 in homozygosity. So we generated organoids from these induced pluripotent stem cells. We validated, first of all, the expression of L-Galles3BP, so meaning the reduction of the L-Galles3BP in the heterozygous point mutation organoids and the complete lack of the protein from the homozygous exon 5 knockout. And then we wanted to see what is going on with these mutant organoids. So first of all, since this individual has periventricular heterotopia, we wanted to see whether there are defects in the neuronal population. What we did first is to count the total number of neurons by FACS analysis, where we didn't observe any statistical differences. However, when we look at the sections of these mutant cerebral organoids, we see that within the ventricular zone of the mutant organoids, we have increased number of new and positive cells, which is neurons that fail to migrate to the cortical plate as it happens normally under controlled conditions. We actually also count the number of ventricles that we find these changes, and we see that this is the case in almost 70% of the ventricles that were analyzed. This was also validated with the exon 5 knockout, and these results show us that we can actually you can use these mutant organoids to model cortical malformations such as periventricular heterotopia. So since, as we have seen already from previous publications, the morphology and the function of the progenitor cells is important for this neural migration disorder. And since L-Galles3BP is mainly expressed in progenitor cells, we then wanted to see how progenitors are distributed in these mutant organoids. So the total number of progenitor cells from our analysis didn't show any statistical differences. However, when we looked closely at these organoids, when we stain the mutant or the control organoids with a marker HOPEX, which is a marker of basal radioglial cells, the cells that lose their attachment and they migrate to the subventricular zone, having only a basal process as shown here and lacking the attachment to the ventricular zone. When we looked at the mutant organoids, we see that these HOPEX cells are there, can be generated. However, they lack, they cannot retract this apical process from that link them to the ventricular surface. So basically, we also count the number of events, and we saw that this is the case in almost 70% of the ventricles, which are the same where we see the ectopic neurons, suggesting that there is a correlation between these somehow misspecified cells that we generated within these mutant organoids, that they fail to retract the apical attachment. We also checked total numbers of progenitor cells within the ventricular and the subventricular zone, and we actually saw the differences that the more progenitor cells are found in the ventricular zone and not in the subventricular zone, suggesting that these mutant cells so there is not an actual number problem, but actually location of the progenitor cells within the developing cerebral organoids. So we then wanted to validate also if this is the case by ectopic overexpression of L-Galles3BP, and to do so, we either electroporated the human version of L-Galles3BP in cerebral organoids or in organotypic slides from human fetal cortices at gestational week 16, and by analyzing these two systems, these human-specific systems. We saw that the number of progenitor cells that are found within the ventricular zone are induced upon L-Galles3BP overexpression, and those that are found in more basal location are increased, meaning the opposite phenotype that what we see in the mutant organoids. Again, suggesting that the position of the progenitor cells upon L-Galles3BP manipulation is affected in this situation. So since the position of the progenitor cells may have to do with their delamination from the ventricular zone, and since these can be changed if the progenitors of the ventricular zone are not attached properly to the ventricular surface, we wanted to see how the apical anchoring of these progenitor cells is in the different systems that we have. So basically, we stained control organoids or organoids where we overexpress the control plasmid GFP with the F-actin, which is a marker of this apical anchoring of the progenitor cells. And we saw the nice formation of this apical belt here. But when we overexpress the L-galus-3BP in these organoids, in the control organoids, we see that there is a disruption of the apical belt at the site of the electroporation and not at the adjustment region or in the control condition. On the contrary, in the mutant organoids, this apical belt is much thicker compared to control, and suggesting that most probably these progenitors are more strongly attached to the ventricular surface, and maybe this is the reason why they fail to retract this apical process that they have. All these experiments were done in vitro in the human-specific system, but in vitro still. So we wanted to see whether this is also the case in vivo. For that reason, we turned into our model system, the mouse system. We performed in vitro electroporation that was already introduced from the previous speakers. In order to overexpress and to introduce, actually, because in the mouse L-galus-3BP is not expressed, to introduce the human-specific form of L-galus-3BP. So then we analyzed the developing cortices, and we saw how the progenitor population, so apical or basal progenitor cells, are distributed in the developing mouse cortex. And actually what we have seen is that when we stained for markers that are found in apical radial glial cells, in the cells that are found mainly in the ventricular zone in the developing mouse brain, such as HOPX or PAC6, we see that there are clusters of cells that are found in more basal locations here. And when we calculated actually the distribution of these cells, we see that these cells are mainly found in this bin C of the cortex, which is a region more basal from bin A, which is mainly the ventricular zone, and more basal from bin B, which is actually the subventricular zone in the developing cortex. And actually, this was also the case for when we count the TBR2 positive intermediate progenitor cells in the developing mouse brain, again having an enrichment in this bin C. And all this data show us that if we introduce L-galus-3BP in the developing mouse cortex, we have the changes in the distribution of the population of this neural population in different areas, and by the formation of this outer subventricular-like zone that is typically not observed in the developing mouse brain. So since we know that these outer subventricular-like zones can be a reason of generating sulci and serine in gyrified species, we wanted to see whether this is the case in this model system. And actually what we saw, even from 3 days post-electroporation, but it was also more pronounced at 6 days post-electroporation or even 13 days post-electroporation, we saw that at the site of the electroporation, we have the generation of this gyri and sulci only at the site of the electroporation and not in the contralateral side of these mice, since the mouse cortex is smooth under normal conditions. This was also the case when we count in almost 60 to 70% of the animals analyzed. And interestingly, these folds were colonized both from upper layer neurons, SADB2 positive neurons, and from deep layer neurons, TBR1 positive cells. Suggesting that this overexpression of L-galus-3BP also in vivo can influence the location of the progenitor population, which then will affect the proper cortical development. So since we know that there are mutations in the gene that cause cortical malformations, we wanted to see whether these mutations may affect also the cortex in vivo. So we generated constructs that have the pawn mutations that are found in individual 1 and individual 2, and we wanted to see whether overexpression of these mutant forms of L-galus-3BP can recapitulate the phenotype that we observe upon overexpression of the wild-type form, meaning the ectopic location of the progenitor cells and the generation of sulci and gyri. However, we actually were surprised to see that not the relocation of the progenitor cells nor the formation of the sulci and gyri was able to be recapitulated from the overexpression of the 2 mutant forms, suggesting that these mutant forms of L-galus-3BP may only function in human and in a system where the mouse is such as the mouse where the L-galus-3BP is actually not there. This mutant form do not actually function. So then the final thing that we observed in the organoids was that the apical anchoring was the one that leads this the redistribution of the progenitor cells in the developing cortex. And then we checked if this is also the case in vitro. In vivo, we use this F-actin as a marker of apical anchoring, and we observed that upon ARHGAPIIB overexpression, we have a disruption of the belt here, where exactly as we saw it in the cerebral organoids. And again, when we overexpress the mutant forms of ARHGAPIIB, this was not, we were not able to recapitulate the phenotype. I'm sorry, I have to plug my computer. Okay, I'm sorry for that. Okay, so since ARHGAPIIB is an extracellular matrix protein and it is known to be secreted, we wanted to see whether the environment to which these progenitor cells are involved, are found, if this is by itself important for cerebral organoid and human brain development in vivo. So what we did is to actually check the proteome of these control and mutant organoids, either the whole proteome or the secretome from these organoids. In particular, we took control and mutant organoids and we lysate them as whole, and we sent for mass spectrometry the whole lysate, or we collect the medium from control and mutant organoids and we send for mass spectrometry. So by analysis, we saw that there are several different proteins that were differentially regulated with this, within these organoids, and within the mutant organoids. And when we perform a geotherm analysis of these differentially regulated proteins, what we observed is actually that the key cellular processes that were affected were processes regarding extracellular organelles or extracellular vesicles or extracellular exosomes, which was actually also a little bit expected since ARHGAPIIB is a secreted protein. We then wanted to see which particular cell type is also affected within these mutant organoids, and to do so, we perform a single cell RNA sequencing transcriptomic in these control and mutant organoids. We isolated the cells, we performed single cell sequencing. We saw that the mutant cells intermingle very nicely with the control ones, and that the pseudo-time differentiation is preserved from progenitors to neurons. And actually, what we saw is that in terms of number of cells, we observed mainly differences in the ventral side of in the ventral identity of the cells, which is actually something that I will not have the time to discuss with you today. But in the cortical region, where we actually are focused on, we see that the main differences is actually in the number of the radial cells. So focusing on this radial population, and we took the differentially regulated genes, only the radial population, and we perform this geotherm analysis again. And again, we see that among the most highly differentially regulated processes, with the highest fold enrichment, are again extracellular exosomes and extracellular vesicles, suggesting that these processes are actually affected only from the radial cells, and this can maybe affect then later on neuronal differentiation. First of all, we wanted to see whether we could actually rescue the phenotype that we observe in these mutant organoids only by putting these mutant organoids, by growing them in a controlled environment. So is the environment by itself important in order for the cortical development to happen in a normal way? So how did we approach this? Is we had control organoids that we cultured in a control medium, and mutant organoids that we cultured in mutant organoids that we cultured in mutant medium, as we had until now. But also we did this switch of the medium, where we had mutant organoids cultured in a control medium that we have isolated from control organoids. So what we did is to analyze this pool of organoids, and we checked actually the two main things that we observed in this in our phenotype, meaning the anchoring of the progenitor cells to the ventricular side and the ectopic neurons. So what we have seen is that, as I told you, the mutant organoids have this thicker belt that we see here. But when we culture the mutant organoids in a control condition, in a control medium, these mutant organoids, they have they have a belt which is as thick as in the control condition, fully rescue the phenotype that we observe. And this is also actually the case with the ectopic neurons, suggesting that only the environment into which these organoids are generated is sufficient to help these progenitor cells to anchor in a proper way to the ventricular surface, and actually to allow the neurons to migrate properly to the cortical development. So since we know now that the environment of these organoids is important for the correct development, and since that from our single cell data we saw that, and from proteome data we saw that the exosomes and in general the vesicles that are secreted from the progenitor cells are actually the most differentially regulated in the mutant organoids, and the fact that we have among the highest differentially regulated genes in our single-cell transcriptome are this family of genes which are called the transpanics, which are actually found, which are actually transmembrane proteins that are found in these small vesicles that are called exosomes. We wanted to see whether exosomes are actually important for this phenotype that we observe. So a brief introduction of what exosome means, since some of you may not know, is that small organelles that are around size of 100 nanometers, that they carry specific cargo such as proteins or RNA, are secreted from cells, and then they can be uptaken from neighboring cells. And according to what their cargo is, they can influence the behavior of the surrounding cells. So basically, the first question that we wanted to answer is whether L-galus3BP is found in exosomes and from the human cerebral organoid cells. So what we did is to take control organoids. We took the medium from these control organoids, and after ultracentrifugation, we isolated the exosomes. We validated, first of all, that these exosomes that we isolate, or what we isolated is indeed exosomes, and to do so, we performed immuno-EM, where we saw that the majority of the vesicles that we isolate are in approximately 100 nanometer size that the exosomes usually have, and that these vesicles express the exosomal marker CD81. And then we took these exosomes, we perform a Western blot analysis, so we lysate them and we perform a Western blot analysis, and we actually saw that L-galus3BP is enriched in this fraction, in this exosomal fraction of the neural progenitor cells. So then we wanted to see whether we can generate exosomes that have different cargos. So if we can generate exosomes that have that overexpress L-galus3BP, or if we can generate exosomes that have the mutant forms of L-galus3BP that the individuals with cortical malformations have. So what we did is to take a neuroblastoma cell line, cancer cell line, these HY5Y cells. We overexpress concepts that express either HA, only control condition, or the wild type form of L-galus3BP, or the two mutant forms of L-galus3BP. We took, after several days, the medium from these cells. We ultracentrifugate them, the medium, and then we isolated the exosomes, and we were able to obtain exosomes that are controlled, normal exosomes, or exosomes overexpressing L-galus3BP, or exosomes that have the mutant forms of the L-galus3BP that we have that we have identified. We first validated that indeed these exosomes express the wild type or the mutant forms of L-galus3BP. And then we took these exosomes and we wanted to see whether these exosomes per se can actually recapitulate the phenotype that we have in vivo in the developing mouse brain. So what we did is to take mouse cortical slices at mouse brain from E13 embryos. We took we generated the organotypic slide. So we cut the brain in the vibrotome, and then we cultured these exosomes in this insert where we have we can put them in a medium, and we incubate them for several days in the incubator, and then we treat these slices with exosomes from control conditions or from the three different conditions overexpressing the wild type or the mutant forms of L-galus3BP. So 3 days later, we took these slices, we fixed them, and we stained them. And what we saw actually by analyzing these organoids, mouse sections, is that at the sections where we have the exosomes that overexpress L-galacthrib-B, we see an increased number of basically located progenitor cells, exactly mimicking the phenotype that we have in vivo when we overexpress the L-galacthrib-B gene, suggesting that this overexpression of L-galacthrib-B that we have. The protein that the gene that we introduce, it can be then generate a protein that it is secreted via exosomes, and then it can manipulate the position of the progenitor cells. And interestingly, what we also saw is that when we incubate these mouse sections with a mutant form, the exosomes that they have in the mutant forms, we actually recapitulate the periventricular heterotopia phenotype that we have, that we saw in the individuals with this point mutation, suggesting that this exosomal secreted L-galacthrib-B can modulate this progenitor population and the formation of the developing cortex. So summarizing, what I have told you today is that we have, I hope that I convince you that the environment in which progenitor cells are found is very important for their function and actually their fate. So we can see that overexpression of L-galacthrib-B in the human tissues or in the mouse tissues, either by introducing the gene or by introducing exosomes expressing the protein, result in the weakening of this apical belt, increased delamination of the progenitor cells, and more generation of sulci and gyri in the developing cortex. While on the contrary, when we manipulate L-galacthrib-B expression either via knockdown or by mutating the gene, we have the opposite phenotype, meaning more stronger attachment to the ventricular zone, misspecified progenitor cells that they do not know where to go, so they stay in different positions, and then this maybe later on affect the proper cortical development, generated malformations such as periventricular heterotopia, and having changes in the cortical thickness and in the gyrification index in the human in these humans. So finally, I would like, first of all, to thank everybody who contributed to this project. I was very lucky to work in my postdoc in Silvia's Capello lab, and many people helped me in this project, all the members of the lab, and especially Rosella, who also contributed to this project. And also all our collaborators, Elizabeth Binder from our MPI, so our director in the MPI, Alex Buffett, who performed the human fetal cortices experiments, Ilka, who did the immuno-EM in the exosomes, our clinicians, Stephen Robertson, Reza Guerini, and Frances Elsmilier, Stefan Sieber, who did the proteome analysis and the whole proteome and the secretome, and also Barbara Treutlein, who did the single-cell RNA sequencing analysis. So I also thank you for your attention, and I will be happy to take any questions.
Thanks, Cristina. Great thought. Is there any question? So I put one in the chat. So from Maria Pignano. Great talk, many thanks. Just a curiosity, do you have evidence of other proteins involved in adherent junctions that are altered in L-galacthrib-B transgenic mouse? Cristina, you muted yourself.
Now, do you hear me?
Yes.
Okay. So just first of all, I wanted to clarify, maybe I wasn't very clear in the talk, L-galacthrib-B is not expressed in the developing mouse brain. So all the work that was done in mouse was with ectopic generation of ectopic induction of L-galacthrib-B. So there is no transgenic mouse, knockout mouse, for example, for L-galacthrib-B. So what we actually checked in the proteome is that we have seen changes in this extracellular matrix, not that much in the adherent junction. So I cannot really answer to you whether there is a specific other specific adherent junction proteins that are affected in these mutant organoids. We also tried to do this co-immunoprecipitation of L-galactose-3BP, and to see whether there are other proteins interacting with L-galactose-3BP and could affect the adherent junction. But also we didn't see any differences there. So yeah, we don't really know if there is any other adherent junction proteins that are affected. But what I can tell you is that in general, from literature, it is known that L-galactose-3BP, upon secretion, it binds with several other proteins, such as also these transponings, and this may affect then the cytoskeleton and adhesion indirectly. All this work was done, though, in the cancer field, in cancer cell lines. So we don't actually have data from in our system, but this is actually could be the case also here.
Thanks. And there is Daniel.
Yes. Hello, Christina. It was a really nice talk, and I really like combining organoids with proteomics, and mouse brain with the neuteros. It was really nice fitting everything together. I have a question regarding the overexpression of L-gal-S3BP in the mouse cortex, where you see this ectopic position of apical progenitors. Mm-hmm. So what happens to the basal ones and to the neurons? Is the layering intact and preserved? Are neurons migrating in a different way? Do you have any information about that?
So I have to say that we actually didn't check how the neurons really migrate. So we didn't do any time-lapse imaging on this mouse. It would be actually very interesting to see whether this is the case. And according to numbers of progenitor cells or neurons, we don't see differences. We actually only see changes in the position of the progenitor cells, and then the formation of this gyri and sulci. So yeah, I cannot really answer to this, but it would be really nice to see, even in 2D, whether we have changes in the migration of these neurons, or at least the speed, or how they can follow a track. So what you saw also with these fibers would be actually a very cool experiment to do and see how they migrate, because this could be, since we don't have changes in the numbers, this could be something related to what you observe also with this flirt system. So it would be really, really nice to actually combine our expertise.
Thanks. I don't have a question, more than a comment. About this mutation in L-gas3BP, do you think that this mutation is having a loss of function effect or more a dominant negative?
Well, yeah. I mean, when we check the protein level, either with stainings or with a proteome, we don't see the protein forming. So we don't detect it, or we detect in a very low levels. So this can be, the loss of function thing, but we actually haven't really checked in a proper way. So it can also be that it is, it has a dominant negative function, but I don't know how we could actually separate these two and answer this question. But what can I say? What I can say is that the protein per se is undetectable, or at least in very low levels in the proteome and in the staining. So most probably this is a loss of function. Mutation, yes.
Thank you very much.
Can I ask something?
Sure.
So there are no questions from YouTube. And I have a curiosity. Any idea how are you mediating the function of your protein? Is there any receptor or other protein that you are suspecting for mediating the effect? And what about integrins, since we heard a lot of previously?
Yeah, actually, L-gal-3BP was shown to interact with several other ECMs, also integrins, and also it is interacting with these transponings that I mentioned to you before, and in particular the CD82. And actually, this is how we enter into this field of the exosome. So we were suspecting at the beginning that L-gal-3BP interacts with the CD82. It was shown already. And actually, this interaction may affect the beta-catenin degradation, which is also can be linked with our phenotype, and the cytoskeleton that we have. From the co-IP that we did, we were not able to detect this interaction, to be honest. But this could be also the fact that we did this experiment in neuroblastoma cells. So this could be also something that it can be specific into this system or something else. But definitely can be this. So these intergreens can be also something quite interesting to dig into it, because ARHGAPIIB is interacting with intergreens, and yeah, I mean, this is something that it's very relevant with this phenotype that we have, and yeah, that would be very good to actually check it in details.
Thank you.
Okay. So if there are no other questions, I think we can close the session of today. And I thank again all the speakers and all of you that were following the webinars. And now, if Ennio De Giudice is here.
Can I say something, Rossella, before Ennio?
Yeah, please, please.
Yeah, I also thank the speakers. They gave great talk, really amazing meeting. And I also want to thank Rossella for putting together all this, because she did 99.99% of the work. So thank you, Rossella, for organizing such a beautiful meeting. Thank you also, the organizer, for being in time. That was incredibly precise. So you were great also in that. And I think that's all from my side. I want to just remember again for the people part of the scientific committee of NBG, please stay for some minutes later. And I think I can give the floor to Ennio De Giudice. Thank you.
I also want to mention all the people that supported this meeting. So you and Simona and Maria Pia and everybody that made it possible. Thanks. Ennio, do you?
Yes.
Yeah.
Okay. Maybe not all of you know about the Neapolitan Brain Group, and I would like to say at the end of this meeting that the main mission of the Neapolitan Brain Group is to put together clinicians and basic researchers, and this makes sense. This is to say that clinicians have to know about basic functions of the nervous system, a basic function of the brain. And if they know about basic research, they are better clinicians. This is one of the main messages that the Neapolitan Brain Group likes to give to all, and in particular to young people, to young doctors, to students in medicine. Tonight we have had three really beautiful talks. We have heard about the role of a specific neuron precursor, the basal progenitors, and in particular we understood how their morphology can affect the neocortex expansion and evolution. Secondly, we also, Daniel Del Toro Ruiz told us how synaptic flirt proteins, so a synaptic protein may modulate the folding of brain neocortex. And finally, and this was also a new idea for us clinicians, we have heard about the contribution of cerebral organoids to the understanding of brain functions and brain disorders. In particular, we heard from Cristina Chirusi about LGAL S3BP, which also is involved in neocortex development and therefore in human brain pathology. So, as the chair people told at the beginning of this meeting, the tradition of the Neapolitan Brain Group has always been that of having an annual meeting in December in presence. This time was not possible, but thanks to Rossella and Elia, we also had, let's say, a kind of mini symposium, mini meeting with very nice speakers and very nice talks. So the last word that I have to tell to everybody is to thank everybody, all participants in these meetings, in particular the speakers, and maybe one of the good things of this very bad event of the COVID-19 pandemic was to bring together people from all over the world and to make us understand that we are the human people. The Earth is something common. There is no sense in dividing races, ideas, religions. We are the Homo sapiens species, and we are all in the same, we share the same conditions. So maybe this could be a good thing from the pandemics, and I hope that this kind of thing may bring together all of us together in presence. So thanks to all. I say thanks to all, and I ask Rossella and Elia to formally end this meeting.
Yeah, as we were already writing in the chat, we thank all of you to participate to this event, and please follow us because there will be new events coming soon. We will prepare a new calendar for new talks in the next months, and that we hope will be interesting for most of you. And I also want to thank again the speaker for the great talks, and I hope to see you soon.
Thank you.
Thank you. Bye.
Bye.
Thank you all.
Bye.
Bye.
Bye. Thank you.
Bye, Cristina. Bye, Daniel. Bye, Nereo.